You're doing a really great job. And is it this one?
Yes.
And pointer again, in case you need it. All right, well, you're familiar with the speaker already, but now she's going to talk about a comparison of blueberry pollen vigor and germination between 4 different cultivars.
Thank you. And welcome back. This is the last presentation for today, for this year. And I'm so grateful you are still here. And now I'm going to talk about comparison of blueberry pollen vigor and germination between 4 different cultivars. And for the background, I want to give you a definition of pollination. Pollination is the transfer of pollen from the male anther of a flower to the female stigma, enabling later fertilization and the production of seeds. And please look at this photo. It's a blueberry flower. And in the center of this flower is the style. And on the tip of the style is stigma, which is the female part of a flower and that will receive the pollen. And around this style, please look at this yellow part. They are filaments. And on the tip of the filament, there are anthers, which contains pollen grains. And in flowering plants, after the pollen grains landed on the stigma, it will develop a pollen tube through the style and finally reaches the ovary. And the sperm cells in the pollen grain will go down through the pollen tube and enter an ovum to fertilize it. And this procedure is usually assisted by pollinating agents like animals, for example, honeybees, butterflies, birds, and also wind, water, or even plants themselves when self-pollination occurs within a closed flower. And in this research, I use 4 different cultivars of northern highbush blueberry. And here I want to help you to distinguish between pollinator and pollinizer. Pollinator is the biotic agent that move the pollen to the stigma. For example, honeybees, birds, butterfly, moths. And pollinizer is the plant which provides the pollen grains. It's the source of pollen for the next generation. And in blueberry, most cultivars are self-pollinated. That means the stigma receive the pollen from the same cultivars, so they can be fertilized. For example, Duke and Draper in this table. And some cultivars are cross-pollinated. That means the stigma should receive the pollen, or we can say pollinizer, from the other cultivar, so they can be fertilized. And this is rare in commercial popular cultivars because to reach a full pollination, these 2 parent plants should be compatible to each other. In most situations, if these 2 cultivars come from the same type of blueberry, for example, they are all northern highbush blueberry, they should be compatible. And the blooming time should be close to each other, so they have enough time to pollinate. And there are also other cultivars in between. Please look at this row in the table, intermediate. They can receive the pollen in the same cultivar to fertilize. And if there are other proper pollinizer, it will have a better pollination. Pollinizer is not necessary, but it's better to have. And usually, a successful pollination and fertilization will result in a seed. And the number of seeds is to some degree relative to fruit size, because seeds release hormones that are early inducer of fruit growth. And this ability is relative to pollen vigor and functionality. By knowing about blueberry pollen vigor and functionality can help us to build a model of effective pollination period. This is the number of days during which pollination is effective in producing fruit. Knowing this, we can know what's the best time to introduce the honeybee hives into the field and take them out of the field, or take other strategies to improve the pollination. And there are few studies examine the pollen functionality, especially in northern high-bush blueberry. There is a study about Pemberton, Berkeley, Jersey, Rancocus, Dixie, and Weymouth high-bush blueberry. And it shows that they have different pollen functionality, they have different germination rate and tetrad abortion rate. But this study was conducted in 1966. Yeah, half century ago. And they are very old cultivars. So we need to know about the cultivars we plant today. So I choose Draper, Duke, Aurora, Liberty to study about the pollen biology. And this is our objective, studying pollen vigor and germination at different temperatures among 4 commercial important high-bush blueberry cultivars to help to improve the current understanding of pollen biology for blueberry grown in Western Washington. And first, we collected the pollen from these 4 cultivars once a week from 1st of May in this year during the whole blooming season. And in total, we collected 2 to 3 times for each cultivar according to the bloom period and weather conditions. And we made pollen-specific media in petri dishes. This media, just like stigma, it will receive the pollen to let it grow, let it fertilize. And we held the flower using tweezers and shake the pollen onto the media. And then we put this media into different incubators under different temperatures, that is 35, 45, 55, 65, and 75 Fahrenheit for 4 days, because the pollen longevity of blueberry is less than 4 days. And we check the pollen under microscope every day. And please look at this photo. There are many tiny white dots that are blueberry pollen under the in the petri dishes. And here I want to introduce a definition of tetrad. It's a group of 4 single cells, and they are immature pollen grains. Please look at this photo. It's a tetrad of blueberry pollen. And you can see there are 1, 2, 3 single cells. And the last one is under this plant because it's a 3-dimensional shape. And after the pollen land on the media, it starts to grow, start to fertilize, and it starts meiosis, turn to a tetrad. And we take photos from microscope. In this case, the magnification is 40. And we randomly selected 30 tetrads in each quarter of the petri dishes. And we measured the pollen germination rate pollen tube number per tetrad, and pollen tube length. Let's talk about the results. Here is the germination rate at different temperatures. The x-axis is the temperature, and y-axis is the germination rate from 0 to 100%. And different colors stands for different cultivars. Blue color is Aurora, and orange is Draper, gray is Duke, yellow color is Liberty. And from this data, we can get the trend lines, which can tell us what's the optimal temperature for a maximum germination rate. If there is at least one pollen tube on the tetrad, we say it's germinated. And for example, please look at this yellow line. This is the trend line of Liberty, and the peak is located at around 55 Fahrenheit. That means 55 Fahrenheit, this temperature is the optimal temperature for Liberty to get a maximum germination rate. So according to this graph, we can know the optimal temperatures for germination rate is 75 Fahrenheit for Aurora, and 55 to 65 Fahrenheit for Draper, 65 Fahrenheit for Duke, and 55 Fahrenheit for Liberty. And now let's check the pollen tube number per tetrad under different temperatures. Still, x-axis is temperature, and y-axis is the pollen tube number per tetrad, and every color stands for a cultivar. Blue color is Aurora, orange is Draper, gray is Duke, and yellow is Liberty. So from this data, we can know the optimal temperature for having more pollen tube number is 75 Fahrenheit for Aurora, and 65 Fahrenheit for Draper, 65 Fahrenheit for Duke, and 55 to 65 Fahrenheit for Liberty. And now let's check the pollen tube length at different temperatures. The pollen tube length, the unit is micrometer, and the result is 75 Fahrenheit for Aurora, and 75 Fahrenheit for Draper, 65 Fahrenheit for Duke, and Liberty. So here is the conclusion. Temperature not only influences the germination rate, but also pollen growth. If there is a low fruit set and a small fruit size, it's not only because the honeybees or other nutrition or soil, it can also because the response of pollen to different temperature. So combine all these results, we can conclude the optimal temperature for pollen germination and growth is 75 Fahrenheit for Aurora, 55 to 65 Fahrenheit for Draper, and 65 Fahrenheit for Duke, 55 to 65 Fahrenheit for Liberty. Now, knowing about this, how can we apply it into the production? Here is the average daily temperature in where we collected the pollen in Mount Vernon, Skagit County. Is in May 2018 during the blooming season. And we can know the lowest temperature is 53.3 Fahrenheit in 1st of May. And the highest one is in May 14, 65.9 Fahrenheit. So this temperature is suitable for Draper, Duke, Liberty to pollinate, to fertilize, but it's still too low for Aurora. So when the temperature becomes proper and the flower is opening, we can introduce our honeybee hives in the field. And when the temperature turns on improper, we can take out the honeybee hives. And if the temperature is still very low, while the flower is already open, for example, aurora in this year, we can spend less money on aurora field because we already know this year under this kind of temperature, aurora won't get a good pollination. And I want to thank again these people who helped me with my research: Sean Watkinson, Huang Zhang, Nadia Boston, Matthew Arrington, and my advisor, Dr. Lisa DeVerter, and my committee member, Dr. Beverly Gerdman and Dr. Matthew Whiting. And thanks to Washington Blueberry Commission for providing fundings for this project. And thanks everyone for listening. Yes.
Great. Thank you again, Weixan. Does anyone have any questions? We've got a lot of time for them if you have them. Awesome. Well, I think that wraps it up. Oh, never mind. Thank you.
Hi, thank you for that. So given that aurora would do better at higher temperatures, can you mitigate that with number of hives per acre or some other management tactic?
In this experiment, we didn't do any other additional management, just common management for the soil and nutrient. Yeah. So, did I answer your question?
So I guess that's the next step from a grower's perspective. Is if he's planted all this aurora, how's he going to manage it to make sure he optimizes fruit set?
Oh, yes. If the temperature is not suitable for aurora to pollination, maybe we can combine bumblebees, because bumblebees is active in a lower temperature and worse weather conditions. So I think this can make up to this shortage. Yeah.
Thanks so much. That was really remarkable work. Very, very well done. The one question I did have is the length of incubation. The temperature will fluctuate through the day. How do you think that might affect some of these results?
Yeah, in the incubator, the temperature is stable, unlike the natural condition. I think it's very hard to control the temperature in outside, and it's hard to mimic the natural temperature. So that's why I just put the average daily temperature here. So this it can be a reference for the temperature. Yeah.
The follow-up question is, how long does it take for it to fully extend? How long was the incubation in the incubator?
4 days.
Okay, thanks.
Yeah, because after 4 days, the pollen is dead because the longevity is less than 4 days. They want growth.
Hi, that was a great presentation. I have a question. I'm not sure if it's with you or maybe someone can help, but I noticed in our fields this year that they were you know how there was a picture up there, and I'm not sure if it was your presentation or someone else's, but where the bee was pollinating legitimately from the top, and then there was some that was on the side that was illegitimate. Why does that happen, and what could be the cause of it?
Actually, this is a very interesting thing, because when I counting the bee visitation, I found in some place, in some fields that with fresh flower, I think the honeybees tend to do the legitimate pollination. to insert the head into the flower to touch the pollen. But if at the end of the blooming season, when the flower is not very fresh, the honeybee is tend to suck the nectar and dig a hole on the petals. And if one honeybee did like this, and the other will following. Yeah, so to some degree, it depends on the period of the blooming time.
Okay, and then following that, so if it does go on the side, then there is no pollination at all, or is there some effect?
I think the pollination will be low compared to the normal pollination events. Yeah.
Thank you.